أهلا وسهلا فيكم جميعا ب Our Event Research Without Boundaries organized by بايس أومادا. بايس أومادا هو نادي البيولوجي الأول بكلية العلوم الجامعة اللبنانية حدث، تأسس سنة 2019 وكان الهدف من تأسيسه نكون عم نساعد ونوجه الطلاب ونعرفهم على الاختصاص. لهيك نحن بنشتغل على 3 نقاط أساسية. أول شي بنعمل أورينتيشن سنوية للتلاميذ بكل السنين. 2 بننظم ورش عمل بتساعدهم بالمسار العملي بعد التخرج، مثل كتابة CV ولينكد إن، وكمان بنعمل شير لفرص عمل وستاج. 3 بنخلق جو علمي من خلال أنشطة علمية، وأهمها المهرجان العلمي الكبير اللي عم نقدر كل سنة نفرجي different sections بالبيولوجي ونعرف تلاميذ المدارس والجامعة عليه. كرمال تكونوا عم تستفيدوا من هالأشياء وتكونوا engage معنا أكتر، فيكم تكونوا عم تعملوا لنا follow على our social platforms انستا، فيسبوك، يوتيوب، لينكدين، وتفوتوا على واتساب جروبس تبعنا. تالت سيشن لليوم حتكون مع جودة صندقلي. هو PhD student in experimental medicine بUniversity of Milan إيطالي، وسيشن بعنوان The Research Continuum Genetics, Neuroscience and the Science Behind the Scenes. Thank you كتير جودة لحضورك معنا اليوم، وإذا أنت جاهز we can start.
Thank you ريم، thank you لكل التيم تاع بايس أومادا للأوبورتيونيتي اللي عم تعملوا للتلاميذ. حبيت لكم تكون شوي سيشن، يعني راح يكون عن بارت صغيرة عن الماسترز، شوي الشغل اللي عملته a little bit after master project، وشوي صغيرة حتى ما كتير يعني أعجاء زدال في شوية وقت أحكي شوي يعني عن الكورنت بروجكت اللي عم بشتغل بالPhD. حتى أعرف شوي عن حالي هيك بطريقة صغيرة. أنا عملت بيولوجي باللبنانية، بعدين عملت ماستر بالجينومكس انهانس بروجرام، وعملت الام2 تيزس بالامريكان يونيفرسيتي باللبنانية، امريكان يونيفرسيتي ببيروت. ومن بعدها ما خلصت حاليا هلا بلشت هلا ثالث سنة بالPhD تبعي باليونيفرسيتي اوف ميلانو. فحتى بلش هيك اول بارت بلا هيك مكتر تفاصيل. حكي بلش دغري بفوت بالمين توبيك اللي كنت عم بشتغل بيزيكلي. ف يعني من وقت ما تخرجت كنت دائما حابب كفي شيء له علاقه بالجينيتكس اكثر، وكنت كثير حابب التوبكس له علاقه بالجينيتكس. فبيزيكلي كانت التيزس تبعي فوكست على جين اسمه 1011 ترانسلوكيشن جين، وهو بيزيكلي it's an epigenetic regulator. فحتى ما كتير هيك اللي ما عندهم كتير فكرة عن epigenetics هعمل introduction صغيرة بس. فلما نحكي عن epigenetics هون usually كل شيء عم يكون في عنا expression أو basically regulation للجينوم تبعنا، بس من دون ما نغير basically sequence. so نحن عم نحكي عن basically كل شيء له علاقة بالhistone basically using كمان في عنا RNAi system رح يعمل كمان regulation either down regulation أو مرات بغير basically expression للجين تبعنا. و mainly an important كمان regulator هو الميثيليشن. فلما نحكي هون الميثيليشن هو basically عم يصير في عنا addition لنيو جروب basically على السيتوزين بالبيز بالDNA تبعنا. so عم يكون في عنا enzymes معينين اللي عم يعملوه هن عم يعملوا addition لنيو basically methyl group على البوزيشن نمبر 5 من السيتوزين. so عم يصير في عنا basically جروب عم يتحول من سيتوزين ل 5 direct. so ما حدا لهيك it's sort of a reversible reaction. so عنا بيصير بهالحالة either passive demethylation or active demethylation. so basically هذا الجروب اللي نضاف عنا هيرجع نعمل sort of removal من خلال either two steps. لما نحكي عن passive demethylation عم يكون في عنا through replication للسيل تبعنا، عم يصير في after one replication للتاني عم يصير في removal لهي basically السيتوزين والميثيليشن. والactive demethylation عم نحكي basically عن enzymes اللي هن حيفوتوا basically ويشيلوا basically الميثيل جروب. ومن هول الانزيمز basically هو ال 1011 translocation. so basically بيصير البروسيس basically شيء موجود بالأورجانيزم من الأين مثل ال Fe2 والأكسجين هتشتغل basically الانزيم و through different reaction هيتحول عنا different substrate. بعدين ما نوصل بالآخر basically 5 كاربوكسيل سيتوزين. هون حيصير في عنا sort of مثل اكسجين عنا للبيز، وهون حيتدخل بيزيكلي السيستم لحتى sort of يصلح هذا اللي عم يصير عنا. So basically الرياكشن حدلا sort of reversible reaction، وهون بيجي دور التات. So التات basically عم يعمل basically هي it's a demethylation enzyme. و throughout the years basically تحديدا من آخر 10 years لهلا this gene has been important ب different basically regulation خصوصا through development since basically عم بغير الexpression الجين تبعنا. In addition كمان TET has other roles basically بالnervous system، خصوصا لو عم نحكي هون different members من TET، لأنه basically TET عبارة عن big family. It's basically 3 members but the whole 3 members كمان فيهم different isoforms. في عنا TET1, TET2 and TET3. TET2 مثلا has been involved in cancer مثل TET1 بينما TET3 كمان عنده role كانت also بالnervous system مثل Alzheimer disease. So basically كل هول الجينز عم يعملوا basically different functions، و at the same time نفس الوقت عم يكون في عنده role important بالdevelopment و differentiation. So basically اللي كان عم يشتغل عليه هو it's not on the human، كان عم يشتغل بموديل هو basically الدروزوفيلا melanogaster أو ما يسمى بالfruit flies basically. And هون حفتك between brackets كسيستم it's an important system basically لأنه من جهة الجينوم تبعه في similarities بينه وبين human genome، ومن جهة تانية كمان it's easy to manipulate لأنه basically الدروزوفيلا عندها life cycle تقريبا بيوصل ل 10 days فمنا مثل other models يعني متطلب كتير وقت. و an important factor كان إنه على الدروزوفيلا كان في just one homologue of this gene. So basically ما في عنا لا TET1 ولا TET2 ولا TET3 it's just دروزوفيلا TET. وهذا الشيء بيسهل شوي دراسة إذا بدنا نعمل ريسيرش أو نفهم أكثر شو عم بيصير بيزيكلي بالفانكشن هيدا الجين. سو حتى هيك نشوف الفرق بيناتهم بيزيكلي مقارنة بالهيومن تات. بيزيكلي الدروزوفيلا تات كلهم تقريبا عندهم the same domains ما عدا TET2 it lacks a specific domain but it's not affecting function تبعها. but more or less basically كلهم عندهم the same domain بعد عليهم substrate ف it's basically functioning as a human TET. بس the main question كانت تبع البروجكت is whether sort of using whole human TETs، لأن إن كان human TET1 TET2 or TET3 إذا فينا نعمل sort of restoration that basically sort of بتكون detrimental phenotypes or sort of inconvenience اللي عم بيصيروا عن الدروزوفيلا through the loss of هذا الجين basically. So basically the project focused على إنه كنا عم نشتغل على sort of a mutation أو تحديدا أكثر على sort of dysfunction بالدروزوفيلا TET. and كان البروجكت basically to try to sort of check إذا by introducing the human TETs شو لما نفوت نحن هو الجين من عن الدروزوفيلا إذا فينا نعمل sort of restoration للphenotypes. So basically the two aims كانوا to sort of study شو عم بيصير basically عم نشتغل ب specific dysfunctional TET gene ومن to try to rescue الphenotypes اللي عنا إياهم. So basically كان الأيم to أول شيء نفهم شو عم بيصير basically بهول flies شو عم بيصير on terms of behavior من جهة تانية كمان on terms of molecular. ف what we did إنه basically we studied the development لهالfly. So by using flies حاملين basically sort of a dysfunctional TET. So basically عم بيصير هون عم نحط sort of using transposable elements basically عم ينعمل sort of an excision وعم تطلع البروتين بالآخر اللي هي دروزوفيلا TET. It's not as well as functional as normal TET. So it's not exactly a complete loss but sort of a dysfunctional protein. What I did is basically I studied basically development. So by doing different behavioral assays, so what we do usually is to study her behavior at different stages. واللي كنا نعمله هلا بالأول إنه basically life cycle تبعها usually 10 days. usually من خلال almost 5 days بصير في عنا what we call instar larvae. شو بيتقسموا first instar, second instar, third instar. تقريبا بعد شي 6 days بيتحولوا لتربة مثل an insect and then usually 2 days later بصير في عنا هالفلاي. وهلا هسه كمان it depends on temperature. It is regulated depending on temperature. Usually it's normally 10 days على room temperature تقريبا 25 ل 20 degree. وفي حال مثلا كانت experiment مثلا بتطلب يمكن وقت أكتر في ممكن temperature عليها بالexperiment كمان ممكن يتأخر الديفلوبمنت، بينحطوا على lower temperature تقريبا 18 ل 16. وكمان عكس صحيح، إذا بدنا نسرع شوية development كمان بينحطوا على higher temperature، بس كلهم هودي كمان ممكن يأثروا depending على الجين اللي عم نشتغل عليه. فبasically by counting number of embryos and analyzing number of larvae اللي عم يطلعوا, what we notice إنه كان في huge basically defects تحديدا بهالنمبر. So كان في sort of an embryonic lethality. مش كل الامبريوز كانوا عم sort of they were not hatching وعم يتحولوا للارفي، كانوا عم بيموتوا على الامبريونيك ستيج. كمان من جهة تانية when we're looking من الستيج هي من اللارفي للادلت كمان same thing. So basically what I did is that we combined basically هيدا dysfunctional tet with basically tet null. So basically a fly can completely tet. So هون we're sort of aggravating the phenotype. And what you can see إنه لما عم يكون في عنا complete loss of the tet كمان عم يكون في very reduction بالnumber of the adult flies. من جهة تانية another type of behavioral assay بينعمل تحديدا هون to check sort of هالlocomotors. So once صاروا هالفلايز جاهزين we can check basically إذا عم بيصير في any effect على الحركة تبعتهم. وهذا الشيء مهم because that عنده رول مهم has been shown تحديدا خصوصا التات 2 والتات 1 عنده roles بالمسلز تحديدا. basically here by doing هذا experimental assay we notice إنه كمان عم يكون كان في a little bit of reduction بهال number of flies لأنه تقريبا كان في عدد منهم ما عم يكون في عنده sort of dysfunction تحديدا. moving on شوية من behavior, want تحديدا to look into the system لأن التات usually is expressed highly in the central nervous system بالbrain وتحديدا on the third instar larvae. usually third instar هن basically صار تقريبا جاهزين نحولوا لبيبا وهون معظم الأورجنز تحديدا كان البرين وكل شيء بنسميه usually disc اللي حينطوا بعدين الlegs أو الeyes. كمان هون صاروا في واحد يعملوا عليهم analysis وكمان mainly the result for that is highly expressed at high level. It peaks تحديدا at high level, so it was important to check the expression on that level. But basically by checking the expression تحديدا لهال form of sort of dysfunctional TET in combination with different markers. So by checking it's an RNA sort of protein which will be important for the neuron development. It's called ELAV. Found by what you can see in red basically is the expression of the neurons in the central nervous system. What we see here is basically central lobe واللي على الجوانب basically بنسميهم usually الoptical lobes تبع البرين. And then basically كل شيء عم يكون تحتك عم يعطي triangle is basically the ventral nerve cord which will later be important for axon development. And basically how we can visualize this specifically with Drosophila which we use is the UAS GAL4 system. UAS GAL4 system is usually present it's basically a protein. So حيكون في عنا fly حاملة هالجال4 مع specific promoter مثل البروموتر تبع النيورونز or any other proteins where we want to check, we want to study. And at the same time we have another fly بتكون حاملة basically the UAS. So basically حتكون هي upstream gene of interest تبعنا اللي هو TET in that case. And then by combining basically whole flies and doing different crosses حيكون في عنا البروجين تبعهم حتكون basically عم تعملنا expression للجين بال specific tissue نحن we want to study. So basically هون what we do is we use usually the green fluorescent protein the GFP and we express basically هالجين تبعنا to sort of see exactly شو عم بيصير and we combine them with different markers. So غير النيورونز كمان we checked into what we call a ventral nerve cord and we check on a protein اسمها slit. So basically slit it's an important for the repulsion and the development of the axons. And كمان we notice إنه في a specific expression عم بتصير basically in what we call the midline glia اللي هو بيجي basically بالنص. It's basically the midline of the glial cells اللي عم يعملوا development on the ventral nerve cord. So another important كمان factor للdevelopment of glial cells هو it's called DREPO. What we noticed here specifically when can see a very interesting expression on what we call the outer chiasm glia. So basically I'm on the border of the optical lobes وعم يكون في a very specific patternal expression ما بتكون موجودة usually وكانت highly expressed using this basically truncated or sort of dysfunctional TED. Moreover كمان than the molecular approaches كمان we wanted to check شو عم بيصير basically on the survival للadults. So غير ال behavioral that we do usually on the embryos or on the level of the ladies, we can also do so many experiments خصوصا على adult stages. So هون what we use is basically called the DAM system. So the DAM system is it's a very nice actual program and a machine where basically flies بنحطوا specific tubes and inside specific monitors and usually بيكونوا connected to specific software and then we leave them usually تقريبا 2 weeks up to 30 days depending on at least enough data لحتى نشوف activity. And what we get at the end is basically Excel sheets, and from these numbers and the Excel sheets we can have so many different basically factors and different numbers. We can check the survival, we can check the activity, the sleep وغيرهم كمان. So what we noticed here is that by comparing the whole flies اللي كانوا حاملين dysfunction بالتد in comparison with wild type flies كان يكون في sort of an hyperactivity, while hyperactivity can mainly present أكتر during الوقت اللي usually flies بيناموا فيه. So basically بكون from midnight till 6 a.m. and then هذا المونيتور basically DAM system كمان بيشتغل مثل بيحافظ sort of على circadian clock للفلايز. So في بقلبه lights بيضووا usually from 6 and then بيطفوا after 12 hours حتى يحافظوا على circadian clock الموجودة. So what we noticed إنه في basically hyperactivity. It was mainly present in nighttime. كمان by comparing the profile and sleeping, noticed the same thing. كمان whole flies كانوا عم بيناموا أقل. كمان خلال the survival لهالadult flies. بينكم ما كان في فرق. كانوا flies they were behaving normally. ما كان في any death or anything occurring on all flies. And the most basically important factor هون كان sort of these flies كان يكون في عندهم هيدا الactivity اللي عم تكون عالية. Moreover كان basically the question الثاني or aim الثاني هو once we had an idea إنه شفنا عم يكون في sort of lethality عم يكون في sort of hyperactivity فكان الايم الثاني is now to check if فوتنا على الجينوم للفلايز حطينا basically الهيومن تيتس هن تيت 1 تيت 2 and تيت 3 شو رح يصير basically للفلايز are we able sort of to rescue الفينوتايب هل رح يصير في عنا any difference. So basically كان عم بيصير خصوصا لما كان عم ينعمل كروس للفلايز we noticed إنه كان عم يكون في sort of lethality اللي شفناها بالأول بس كمان هي confirmed لما كان عم ينعمل كروس مع الجي اف بي and then basically الفلايز اللي homozygotes. So basically بهالحالة كان عم يكون في عندهم 2 copies من ال dysfunctional TET ما كانوا عم بيعيشوا basically which was further confirming the results شفناهم قبل. So basically هم كانوا عم بيموتوا على embryonic stage. So we focused تحديدا على stage and can basically aim إنه to check is that sort of after introducing the human TETs we'll be able to rescue. So محفوظ كتير بالعجقة هون but just to show you a little bit there's different steps خصوصا in genetic crosses لنوصل basically لل final lines اللي نوصلهم to introduce basically human TETs depending on different markers موجودين على الفلايز بيساعدونا ننقي بين fly والتاني. And then actually at the end there was no rescue which is normal. Data is not always positive but there was no rescue of the lethality لأنه حتى after introducing these basically human genes ما كان عم يكون في rescue لlethality. كان basically كمان عم يرجعوا عم يكونوا homozygotes in presence of the human TETs ما كان عم يعمل أي effect. But this doesn't mean إنه ما عم يكون في effect on the genome of the fly because at the end عم نفوت بالآخر a human gene بقلبه. بس for basically الaim that is we did not go inside more molecular approaches. The aim was تحديدا who we wanted to check is من خلال how the homozygotes basically embryos if we're able to do sort of a gene expression analysis by doing some PCR or Western blot. But the issue was إنه ما كان عم يكون في أصلا larvae لأنه كان بيموتوا على embryonic stage which a little bit made it difficult بهالحالة لأنه it's not always easy to do a dissection of the brain من هول embryos. So basically the project did not stop here. بعد التيزز a little bit I continued working a bit on the project and the focus تحديدا كان أكتر شوي على الهيومن TET3 وتحديدا focused on the glial cells. Basically الفوكس كان على human TET3 لأنه by bioinformatic analysis it was shown that human TET3 is the closest one to the basically the Drosophila TET, and most of the studies always show إنه دايما في similarities بيناتهم in terms of function وحتى in terms of structure. So what we did is, we expressed basically the gene in the glial cells using the repo, and then we tried to sort of do the similarly behavioral analysis. We did not notice any different changes, but what we basically noticed is that by expressing هذا الجين بالouter glial cells اللي كنا فرجيها قبل بالسلايد اللي قبل, so basically بهالباترن موجودة, we noticed لما عم نعمل basically down regulation, so هون كمان similarly by using ال UAS GAL4 system, بس هالمرة بدل ما يكون في عنا GFP عم نستعمل basically هون UAS DTET RNAi, so basically عم نعمل down regulation لهالتت بقلب هود specifically tissue. we noticed إنه كان عم يكون في a little bit of reduction بالsurvival للembryos, so كان في عدد من الembryos ما كان عم يعطينا adult flies. كمان من جهة تاني we try to knock down the TET, the Drosophila TET in presence of the TET3 نشوف إذا عم يكون في differences, بس كمان same thing when looking into the behavior ما كان في any specific or significant changes. Now أكيد this does not mean إنه ما عم بيصير شيء تحديدا. we're looking at the end تحديدا على behavior أكتر. if we want to look more شو عم بيصير ممكن على methylation pattern, since the TET is a demethylase enzyme. هون we can go for mainly probably higher techniques مثل chip sequencing or a type of sequencing in order to confirm إذا عم بيصير في any changes on the methylation pattern أو على البروفايل تحديدا. What I did after the thesis and after the work تبعي كمان I was working a bit on the project and some side project تحديدا, also focusing on the DTET. I'm not gonna share كتير معلومات عنه at the moment because I can't, but we're focused تحديدا on a project تحديدا on the TET2 تحديدا, which is an important one in the pediatric cancer, and focused أكتر على specifically على الهistone mutations. So by focusing on the TET2 كان في basically we try to express هذا الجين تحديدا هو mutation على pediatric cancer in the eye of the Drosophila, since it's a little bit easy to see شو عم بيصير, ما في داعي نعمل dissection or anything. And I noticed when we were expressing this mutation كان الفلايز عم يكون في عندهم a smaller eye phenotype, و at the same time كان يكون في sort of a change in the form of the eye. So الانتينا مسمى تبع الeye ما كانوا normal as usually, كان يكون في presence of the methylation على الهستون and also mutation على ال TET2, كان يكون في a specific change بهالeye. More importantly, what we noticed إنه when we overexpressed the Drosophila TET كمان هون using same thing, the UAS GAL4 system in the eyes, you know, the overexpression was leading to a completely different phenotype. Most of the flies كانوا يخلقوا without eyes at all. So again showing role at detecting كان بالnerve system أو حتى بالeyes. Another side project I was working on, it was completely not related to DTET, but still regarding neuroscience and moreover كمان the central nervous system. It was a collaboration كمان with a university in Sweden. So the project can focus على anaplastic lymphoma kinase or ALK. And basically what that group did is that they discovered sort of a new neuropeptide that they called Sparkly. And what they wanted to check basically is شو عم بيصير on the behavioral levels. So what they did is that they generated some flies having specific mutation on specific exons, in addition to some loss of function of the mutation. And they send those flies. What we did basically is that we analyze them through the Drosophila activity monitor, DAM system, and we noticed there were so many differences on the term of the activity. So basically Sparkly, the neuropeptide الجديد, can sort of كمان having different patterns. So من جهة when comparing it to normal flies كان عم بيبين a pattern that we do not usually see on the flies. So usually flies قبل ما يضوي الضوء بيصير في عندهم a sort of what we call an anticipational activity. ومن جهة تانية قبل ما يطفي الضوء قبل ما يناموا basically بيكون في عندهم sort of anticipation, but in that case كانت anticipation عم تكون sort of dysregulated. And same thing by checking other factors and other parameters, we noticed there were so many dysregulation خصوصا عن basically this mutation of the sparkly. حتى ما كتير ضيع وقت, I will not go through much of these data. I'll skip a little bit to what I'm currently working on at the moment, which was not actually easy because after 20,000 applications and after so many rejections and interviews, بآخر 2022 I was accepted into the program in the University of Milano. I'm currently working. I'm still working. I was interested always to work in neuroscience rather than genetics. I made sure the project that I was choosing can, through my application, whatever I was applying for, always something related to what I like to do. I was always interested in the neuroscience field or genetics. What I'm basically currently working on is a sort of a neurodevelopmental disorder, which is called Rett syndrome. It's sort of related to autism. At the moment, it's not classified as autism. It's classified as a severe actually neurodevelopmental disorder. And what's actually interesting, بآخر actually 90s أو 2000, basically the main genetic driver of this disorder was discovered by a Lebanese scientist, and she discovered basically the gene, which is basically the methyl CpG binding 2, which is also an epigenetic regulator and has a very important role in the brain development. And it was discovered as the main cause of gene health disorder. So basically, this disorder is mainly classified as a severe neurodevelopmental one. It's basically progressive disorder. So basically, symptoms they start to be aggravating throughout the age, and it's mainly prevalent in females. So usually females بيخلقوا normally until تقريبا almost 8 months ل 12 months old ببلش عندهم sort of symptoms, and the main affected basically function حيكونوا mainly عن intellectual disabilities, كمان الموفمنت طبعا, and progressively عم يتغير basically كل هودي وعم بصير في عندهم loss of even ability to speak حتى or moving. So what's important to notice إنه MECP2 is mainly mutation in this gene who basically is leading لهال disorder. اللي عم بصير basically هون إنه أحيانا mutation in MECP2 is leading to dysfunction on the level of the development of the neuron وتحديدا على synaptogenesis, and synaptogenesis basically اللي عم تكون important يكون في عنا basically regulation and transmission of the messages. And the mouse model is basically هون the model تبع disorder. So it was shown تحديدا by introducing and creating mouse model حاملين هال disorder. كمان عم يكون في عندهم same thing مثل human, عم يكون في poor grooming and as well كمان reduced mobility, at the same time lifespan تبعهم عم يكون مثل patients affected. But basically the main aim of the project would sort of find a new gene, تحديدا what we call a modifier gene. لما نحكي عن modifier gene, basically genes that are not causing the disorder but are sort of عم يعملوا مثل collaboration مع main gene هو MECP2, like they're either aggravating the disorder or ممكن عم بيحسنوا disorder. The gene that I'm focused on is called the hypocalcin-like 4. And why basically this gene was chosen? Because literature from RNA sequencing data from the lab, it was shown that the gene was always downregulated in the brain of rat patient. Also in the brain of mouse model حاملين or that have this disorder. Now the problem is this gene is sort of belonging to what we call the dark genome. So the dark genome هو basically نسميهم T-dark. They are genes that don't have specific function. We don't have much information عنهم. And there's not enough حتى مثلا materials, antibodies, or papers that confirm a role basically gene, which is basically known. The gene belongs to a family of calcium sensors, on the level of the neurons. So basically, calcium is important in the transmission of the messages. But beyond that, there's no information. And here comes basically the project and the aim of the PhD. So basically, main aims of the project is to check where is this gene expressed, شو on the level of the neurons, and من جهة تاني to check and to identify basically the main function of the gene. Hopefully later on, in case we find some important actually data regarding this gene, to sort of try to do a rescue experiment and to try to introduce this gene in mouse model and check what is exactly happening on the level of Rett syndrome. Basically, what I did is to sort of study the expression of protein. So by checking expression on different levels on different ages, تحديدا بالcortex والhippocampus, because usually هن main areas of the brain where usually Rett syndrome عم يعمل effect أكتر شيء. So by checking expression هون by taking the brain from the mice and checking different ages, تحديدا P0, P7, P20 until 70 days of age. Why this was important? Because mainly, as I said, disorder is progressive. So throughout the age, the symptoms, they are aggravating. We noticed that there's an increased level. So basically the protein, basically, it's increasing throughout the age, similar to MECP2. MECP2 is also an important protein on the brain. It is expressed mainly in the brain throughout the development, but this one is showing sort of an increase in the expression. كمان, to confirm this, we moved in vitro and we checked on the level of the neurons, and same thing. We noticed إنه كمان عم يكون في increase of the protein levels. What was also important to check is to see إذا كانت الexpression specific on the brain وموجودة كمان outside the brain. And by checking in vitro the astrocytes, and by checking different organs from the mice, the lungs, the kidneys, the spleen, we noticed, and basically the protein is just mainly abundant in the brain with very, very low levels in other organs, and حتى astrocyte. It is expressed and highly enriched on the level of the neurons. And هون بيطرح السؤال again, what is basically this protein is doing on the level of the neurons. Moreover, by comparing the levels of the protein in comparison to wild type and in comparison to mice that have basically knockout of the MECP2, so basically they're basically Rett syndrome mice, they are recapitulating the disorder. We notice إنه كان في downregulation of the protein specifically at older stages, so P60 and P40. So when we're talking here about ordered stages, هنا نحكي أكتر عن symptomatic stages that disorder. كمان same thing in vitro, by comparing كمان different levels of the protein on different ages of the neurons in vitro. We noticed إنه كمان كان في downregulation of the protein levels at also higher and older neurons. كمان by comparing also what we call the synaptosome fractions. So only by using the brain from the mice, what we do is we extract basically the fraction يلي حاملة السينابتوزوم كمان مع other membranes to see basically شو عم بيصير basically on the level of the synaptosome, since these are affected disorder. We notice by downregulation of the HPCAL14 on the level of these basically fractions. Moving on from then, having an idea a little bit شو عم يصير in the brain and where, when is this protein expressed, كان السؤال where is it actually expressed? Is there any specific expression by neurons? So what we check by immunofluorescence is the expression of the protein, and only since HPCAL is sort of, I said, a T-dark gene, it was not easy antibodies, since it's not a very well-known protein, and most of the antibodies are coming from companies that were not known or that they did not test it that much. It was a little bit of a struggle, but at the end we're able to confirm the expression, and as you can see, can see sort of an expression similarly in all the pattern of the neurons and on the level of the soma, as well on the level of the dendrites. To sort of confirm شو عم يصير by using specific markers, want to check شو عم بيصير basically on the synapses. هون by using specific marker مثل السينابسين we check on شو عم بيصير فيه on the presynaptic compartment تحديدا. And what we noticed is كان عم يكون في basically an enrichment. So as you can see here by comparing synapsin with the expression of the HPCAL4 كان يكون في high enrichment of this protein on the presynaptic compartment. And what we usually do is that we sort of treat those neurons in order to check شو عم بيصير on the normal levels, and at the same time شو عم بيصير فيه لما يكونوا neurons stimulated sort of KCL, and بيتحول basically a sort of stimulated neurons. And similarly we notice إنه كمان يكون في an enrichment on the presynaptic compartment. Moving on from basically after having an idea where and when this protein is expressed, now we're trying to look أكتر using different markers. We're trying to look أكتر to see with post-synaptic markers شو عم بيصير تحديدا. An important aim is to sort of تحديدا identify a function مش بس where is it expressed, when is it expressed, but شو عم بيصير basically. So are the proteins that are interacting with HPCAL4. Once we have an idea on whole protein, it will be easy to see the pathway اللي على فيها protein. And basically this was done by doing what we call a communal precipitation in HPCAL4 and using specific magnetic beads and using the lysates that we're using. In this case, we're using lysates from the mice brain تحديدا. What we do is that we put them in contact, and after several basically washes and using specific magnetics racks, we get basically our lysates, which will be عبارة عن the proteins that have interacted with the antibody. And we send those basically samples to mass spectrometry to check the results. So based on the results that we got, we were able to have a very huge list of interacted proteins. And based تحديدا على the significance in can p-value, log-fold change, as well as the function of the proteins, we selected a list of 7 to 10 proteins in order to validate those and to confirm them. So basically, after having these proteins, what we do is that we do another immune precipitation in order to confirm these results. And we were able to identify and to confirm sort of some of the proteins so far, still a little bit in progress the same. And most of the proteins that we identified can be either related to sort of synaptic transmission, as well as protein reliable also calcium signaling. And an important one, some proteins were important for epilepsy. So كانوا epilepsy, and we're talking epilepsy here. Epilepsy is sort of another symptom of the disorder of the Rett syndrome. Therefore, hopefully, once we have a clear confirmation of this protein, we're trying now next to sort of confirm a little bit on interaction. So basically, by having an idea of those pathway throughout, again, communal precipitation to confirm more protein and to go in depth on interaction. Moreover, by doing now an aim that we're starting now is to check by electrophysiology, شو عم بيصير basically on the level of the neurons. Either by downregulating or by overexpressing header genes using specific lentiviral vector, شو عم بيصير basically on the level of the neurons, and therefore شو عم بيصير عنا basically hopefully after having some data to check if we can restore basically or have this gene introduced in mice depending on the data. I hope that was clear. If you have any questions, I'm here, and thank you.
Thank you so much جودت على البرزنتيشن. يعني أنت مجال شغلك في بقلبه كل شيء تقريبا جينيتيك ونيورو دروزوفيل كله. يعطيك العافية.
يعني ما هي تقريبا هذه التشينج was not easy from organism للتاني لأنه completely different topics, but yes, still related to neuroscience and genetics.
يعطيك العافية. إذا حدا عنده أي شيء تفضل. علي بزري تفضل.
Hello Jawdat. أول شيء
علي.
very hello. It's been كمان while we used to go together على الدبية إذا تتذكر. It's been a few years since then.
Yes, ما بدي أتذكر بس yes. Yes.
So first of all, amazing talk. To be honest, I really like the contrast between the two different إذا بدك research disciplines اللي أنت فعليا اشتغلت عليهم, as Reem just pointed out. I had two questions actually. أول شغلة بالنسبة لقصة going back to the Drosophila story. One of the points I had written was you mentioned that Drosophila can function, or Drosophila TET عم نحكي specifically, can function as a human TET. أول شغلة, to what extent can the results that you observe within the Drosophila be إذا بدك extrapolated to what we see in humans? So one of the main things, or two main things you mentioned, were regarding the motor skills. So to what extent would a mutation of X, عشان انت مثلا you mentioned that TET3 is the one that is most closest to the human TET, right? So to what extent, for example, would an X mutation within TET3 that you do in Drosophila be directly translated to that in humans? شغلة. Or maybe as a follow-up to that same question, عشان الحين نكفي على الDrosophila story, you mentioned the same thing about you mentioned regarding the circadian rhythm. I believe it was slide 14, and you mentioned that the TET mutants they were hyperactive in the nighttime. So what can you, again, if we want to extrapolate this data to humans, how can that be extrapolated? What can we expect?
Actually, thank you for the question. Regarding أول point سألنا عن TET3. في مظبوط, كان basically main hypothesis إذا بدك تبع thesis, since usually in articles or by bioinformatic analysis can usually TET3 is the main one which is closely related للTET. فكانت الhypothesis تبع thesis okay, TET3 will rescue, هنشوف rescue, we did not see anything. But again, we're looking at mainly behavioral phenotypes علشان بيصير in depth تحديدا, and how it can be translated. It will be interesting one. Actually, my colleagues اللي عم يشتغلوا على project or similar project, they're going into different now approaches, مش بس يعني behavioral. So what they're studying now using تحديدا going more into إذا بدك عم نشوف methylation pattern and trying to find تحديدا binding partners تبع التت in the Drosophila. So basically أنت لما تعرف بهالحالة which basically proteins أو شو بصير basically binding partners, and it will be easy to have an idea on function تبع الجين, which will be easier to translate into human. Regarding any points عن hyperactivity, how it can be translated, حلو هون actually this type of experiment is you can get from it important data خصوصا تبع activity. Regarding the humans, basically the genotype كان بيشتغل فيه وكان عنده sort of a truncation, so ما كانت loss exactly, بس كانت a sort of TET that was not functional, so it was not functioning properly. فهون sort of, at least from my hypothesis, it could be related to sort of a hypermethylation profile. So it's usually that it's a demethylase enzyme. فلما ما عم تعمل sort of the regular يعني function, لما عم تعمل هذا الreaction تبع demethylation, it could sort of be leading to maybe hypermethylation profile that might be sort of affecting in كان basically behavior اللي عم نشوفه. So again, ممكن probably يعني my hypothesis it could be to the methylation. So if we go in depth تحديدا, it could be mainly the main cause اللي عم يعمل هذا الeffect اللي عم نشوفه.
هلا you mentioned that إنه studying that, I mean what you said at the end, maybe taking that and forward, going forward with it. You mentioned that other colleagues, did you consider doing the checking for the patterns, because you briefly mentioned it during the talk?
True. يعني بتذكر I think بالdiscussion it was just my thesis, okay, maybe if we check methylation or stuff like this, because during يعني هو 7-6 أشهر بالthesis, like it was just mainly a little bit of molecular and behavioral. But yes, mainly by going more in depth with higher techniques, it's easier to understand the questions that might help us to understand the role of the gene.
Cool. So I'm sorry, I'll add one more question to the list, so it's going to be two more. بالنسبة, you mentioned كمان at some point, I'm sorry, I don't have the slide number written, but you mentioned that Drosophila that were lacking TET actually had, that were lethal at the embryonic stage. And if I've understood this correctly, you were also struggling to actually study them because when crossing, when you have everything dying before they actually grow up, it's actually a problem. So, but what I understood is that so how did you eventually try to introduce the human TET? Did it just fail at completely انه انت حاولت تضلك تعمل هدول but they kept dying so you just gave up ولا كيف؟
عشان كده حطيت بس هيك لأنه the food fuel it will be hard and it's so much genetics and crosses. But basically, so we have a line العناية and we have another line حامل human TET transgenes. So by doing her crosses with specific, because the results عندنا specific like markers بيساعدونا نعرف الprogeny. So it's a little bit, it's not 10 days يعني بتاخد قصة شهرين لحتى يطلع بالآخر اللاينز اللي بدك إياهم. Basically I was looking at the progeny لأنه basically في progeny بيكون في عندهم the GFP and then in the same progeny في منهم ما عندهم GFP, so بيكون في عندهم basically homozygous transgene lymphocyte. So كان شو basically هذا ال basically report اللي هو عنده هال dysfunctional TET لما بيكون homozygous وما بيكون في GFP. What I noticed إنه على الحالتين in case before introducing هيدا line, لأنه basically كانت البلان إنه by doing PCR basically on the brain from the larvae تحديدا على هيدا اللي هو homozygous line to check the expression للتاتش شو عم بيصير. Because it's not easy to do it لما ما يكون في عندك homozygous line. But at the end ما كان عم يطلع line أبدا. So that's why I can focus على الpoint إنه okay, let's check now if we introduce now a human TET, are we going to be able to see على القليل هول larvae عايشين, but it was basically the same thing.
All right. And now maybe to the end. هون عن جد بدك تتحمل شوي عشان شوي هيك يعني بتحرقص لما يكون في بروتين interaction. فعليا عشان هيدا something we've struggled a lot with in the lab.
Same.
So here you mentioned mass spectrometry. Let's just say it's more misses than hits, to say the least. But one of the major things that, for example, we encountered, and I'm very curious how you decide or how you plan to overcome this. Something that we saw was في اثنين proteins نحن عم نشتغل عليهم. We almost expect them to be interacting, and whenever you do IPs, it's virtually impossible to detect that. When you do pull downs, it's very, very difficult, but upon like tons and literally months of optimization قدرنا نفرجي هيدا الانتراكشن. هون انت اللي كنت عم تفرجيه انه you did the IP. One of my questions would be طب انت شفت X number of interacting partners or potential interacting partners by mass spectrometry. I think we have to use potential as with very with confidence. عرفت كيف؟
Yes.
بهيدي الحالة what assuming you do not see an interaction لما انت تعمل IPs, how can you nullify the fact that this is a false negative انه it's simply not sensitive enough؟ عشان اكيد نحن بنعرف انه ال IP you're using an antibody detected and antibodies are huge. فانت عندك one of the possibilities انه simply the confirmation تروح تسكر the possibility of the interaction of other partners. So how do you decide or how do you plan to tackle that sort of an issue to avoid false negatives in your data?
إذا فهمت إنه قصدك بعد ما يطلع عنا الريزلت من الماس سبيك إذا ما كانوا interacting، هل قصدك عن إذا بعد ما عملت ال IP again is what you mean؟
يعني إذا أنت عندك كان hit بالماس سبيك وبعدين أنت عندك false result أو negative result بال IP, how can you basically okay, I mean it could be that it could be a mishit which is completely normal in mass spec for anyone that has ever done it, you know this really well, right? بس الفكرة إنه أنت كيف فيك تجزم عشان ال IPs while good they're not the most sensitive. how do you plan to tackle that?
هذا بلانز الحلو انه هلا السكند يير كانت كلها بيزيكلي على قصه الاوبتمايزيشن. it was just I'm trying to confirm 2-3 بروتينز. حتى from on your point تحديدا from the mass spec هون عم شوف كان في عندنا 5 replicates but at the end we used the 3 replicates لانه as we said كان في 2 of them that have so much negative results. هلا that's why يعني لما عم بعمل confirmation alone using QIP it was not the same تقريبا protocol that I used for mass spec, so غيرت either بالwashes و so much stuff حتى we considered even trying to use new beads. فat the moment يعني عم بلش my third year and there's still some protein trying to be validated حتى just we still يعني in September بلش and I'm validating some proteins from last year. Some of them are يعني 50% validate لأنه الساعة مثلا بيطلع إذا بدك we're seeing positive results, we're seeing البروتين بالIP, and then when you want to confirm it again حتى okay again we're seeing مرات يمكن عم يطلع عم بين من الاشو بالنيجاتيف كنترول عم بين في كونفرميشن، فهيدا كمان عم بكون ايشو، عم بكون في عنا عم ببين عنا هالبروتين بعدين عندك بالنيجاتيف ريزلت بيزك بالنيجاتيف كنترول سيم ثينج. فا اوكي كيف فيني اعرف انا اذا 100% interacting or not. So through hard 1 year of co-immunoprecipitation and optimization, actually at the moment like زي ما بدك يعني we're considered changing from the brain, so lysates حتى غيرت يعني جربت lysates from the neurons, but المشكلة you have some proteins that are not expressed in vitro, and then trying to move now at the moment what we're trying to do is to overexpress. So we bought specific إذا بدك vectors of those proteins. يعني جبنا كل الانتيبوديز اللي هو الليست، حتى الانتيبوديز نص مش but at the moment now we're moving into getting those vector and overexpressing them in vitro, and then after overexpressing them كمان doing the IP basically on the lysates from those. So hopefully fingers crossed remaining potential ones to be confirmed by that way. So it's a little bit يعني a struggle لأنه moving from the brain where you have your mass spec results to now moving to in vitro, but still just at least to validate the results.
Very, very cool. Good luck.
Thanks. Good luck to you too. Thank you for the questions.
إذا حدا كمان بيحب يسأل تفضلوا. أوكي يبدو ما عاد في أسئلة. شكرا كتير جودة للبرزنتيشن ولوقتك اليوم. وبكرة آخر سيشن حيكونوا الله يعافيك. فبنصحكم ما تفوتوا الفيرتشوال لاب تور، وغير هيك شكرا لحضوركم جميعا، يعطيكم العافية.